In this lecture, I'm going to provide an overview of the structure of exocrine glands in the body and some indication of the function of these glands. But I'm also going to deal with them in more detail, where appropriate, when I review the organ systems in later lectures. Exocrine glands are very important in a number of different organ systems, and it's important that first of all you understand what a gland is and know that basically these glands form two major categories. They're either serous secreting or mucus secreting. You need to also understand the way in which glands secrete the components that they have made and how we classify glands or name them. And most importantly also, it's important to have some understanding of the duct system of these glands, because these ducts within some glands modify the secretion product produced by the secretory cells. Now, there are a number of different types of glands. As I explained, I'm mainly going to concentrate in today's lecture on the exocrine glands. They're glands that synthesize, then secrete their products onto the surface. They do so either directly, such as we see in goblet cells lining some epithelia, or they secrete their product via a tube called a duct or a conduit. And as I mentioned earlier, some of these ducts modify the secretion of the glands. There are also endocrine glands. These are glands that synthesize their products, but then they secrete them into the extracellular space around the glands. Because during development, when the epithelium invaginated into underlying connective tissue, the connection between the surface of the epithelium and the secretory invaginated epithelial cells is lost. And so there is nowhere for these glands to secrete their products. So therefore, they secrete them into extracellular space, and then those secretory products move into the bloodstream and delivered to target organs often a long way away from where these glands actually synthesized the material. Another way in which glands are classified or named is because often some cells can secrete products that affect their neighbours, and these are called paracrine glands. These paracrine glands secrete their products into the underlying interstitial space, and that diffuses to nearby cells and affects their function. We'll deal with those in another lecture. So basically, the types of glands are really classified on the way in which they secrete their products and also the way in which those products reach their target tissues, either through a duct, as in exocrine glands, or via the blood in endocrine glands, or just via diffusion along local interstitial space to affect neighbouring cells. It's important to understand how glands release their secretory products. And as I said earlier, in this lecture, we're going to concentrate only on exocrine glands. Well, there are three ways. In this diagram, there are three representations of the way in which exocrine glands or secretory cells secrete their products. Firstly, there is merocrine secretion. Now this secretion, this form of secretion, this mechanism of secretion is probably the most common. The secretory cells make their products and they store their products within little membrane-bound granules at the apex of the cell. And when they're stimulated to secrete, these little granules containing the secretory product moves to the apical surface of the cell, the membrane fuses with the cell membrane, and the product is released into the luminal space by a process we call exocytosis. So that's merocrine secretion. Another way in which the cell gets rid of its products is by apocrine secretion. Apocrine secretion is where often the secretion product is stored at the apical surface, but then that apical surface or a component of that apical surface breaks off. And so what is released into the luminal space is the secretion product, but also a small part of the cytoplasm of the cell. And this is common in some of the glands in the eyelid and probably in the mammary gland. And the remaining form of secretion, the remaining way in which cells release their product, is called holocrine secretion. This is where the cell synthesizes its product, accumulates the product within the cell cytoplasm, but then undergoes programmed cell death. So the whole cell is lost into the luminal space. Holocrine secretion we will see when we look at skin, when we look at the sebaceous gland in skin, where holocrine secretion occurs. Now, there are different ways in which exocrine glands are named or classified. First of all, if you look at the epithelial surface shown on the left-hand slide, the image of an epithelium in the gut, you can see single glands or single secretory cells called goblet cells. They look like a wine goblet. They're shaped like a wine goblet. And these are called goblet cells. They secrete mucus. They're an example of a unicellular gland, a single cell sitting in an epithelial surface all on its own, although there are clusters of these cells all the way along the epithelial surface. If you look at the image on the right-hand side, this is an example of a multicellular gland. Here is a sheet of epithelial cells that are secretory, forming a number of layers. And if you look very carefully at this stomach mucosa, these layers of epithelial secretory cells, you can see that they stain differently. The surface ones are very clear staining. In the middle, you can see some cells that have a pink, reddish stain. And down the base of these epithelial layers of cells, you can see bluish stained cells. And that indicates that in this sheet of secretory epithelial cells, there are 3 different sorts of secretory products. So that is an example of a multicellular gland. Now I want you to remember the word mucosa that I've shown you on this slide, stomach mucosa. The word mucosa is a term I'm going to use in a number of lectures, particularly when we look at the organ system. So please remember that word, because later on towards the end of the lecture, I'm going to explain what a mucosa is. I won't do it now because it's more important and more appropriate towards the end. But just remember that word mucosa. Okay. These multicellular glands can get far more complex than just a sheet of cells that you saw in that stomach mucosa. They can acquire a duct. Rather than just secreting onto the surface like a goblet cell or like these cells in the stomach mucosa, during development, when the epithelium invaginates into the underlying lamina propria, the portion that connects the invaginated epithelial cells to the surface is retained. And that forms a conduit or a duct. The deeper epithelial cells that have invaginated into the lamina propria become the secretory cells, the busy cells that make the secretion products. And so we can further classify multicellular glands of having ducts. And if the ducts don't branch, we call it a simple gland. But if the duct branches, like you see in the 3 diagrams down in the bottom of this slide, we call it a compound gland. Now, the classification can go further if you look at the nature or the shape or the structure of the secretory product. And I'm not going to go through all the names listed here. And I certainly don't want to emphasise these names, or I certainly wouldn't expect you to be able to recall these names, because in reality, when you look at sections through multicellular glands, it's very difficult to appreciate the three-dimensional structure and therefore be able to name the glands like you see listed here. It's very different, though, to needing to remember the way in which epithelia are named and classified. I mentioned earlier that there are some cells, such as the goblet cell, that secretes a mucus-type product. But there are also other cells that secrete a serous-type product. And I'll explain what those two types of products really are in a moment. In this slide, you can see two images, two histological sections taken through two different exocrine glands. One is dominated by mucus-secreting units. The other is dominated by serous-secreting units. And you see here, they stain very differently. In the case of the mucus-secreting unit, or mucus-secreting cells, the bluey tinge or the light staining is due to the component of the secretory product. Mucus-secreting cells or mucus-secreting units, we call them acini as well, which I'll explain later on, they secrete proteins that are glycosylated with anionic oligosaccharides. And because of that, these proteins, these secretory products, are water-soluble. So depending on the fixative you use, often these secretory products are lost from the cell. Here, in some way, they've been a little bit retained. On the right-hand side, you see serous-secreting cells. These are proteins. And if the tissue is being preserved very well, you can see little tiny granules containing these protein secretory products stored in small vesicles at the apex of the cells. Often you'll see the rest of the cell, the rest of the cytoplasm, basophilic, and this reflects the enormous protein factory inside the cell that's making the secretory product. Go back to the mucus-secreting unit. The cells there have nuclei that are squashed against the base of the cell, so they appear flattened. The serous-secreting cells have nice, rounded nuclei, and often that's a good indication or a good way to identify the difference between mucus-secreting cells and serous-secreting cells. Sometimes a gland can have both secretory units. Here you see a serous-secreting unit, very pink staining. Here you see a mucus-secreting unit. And here's an example of what I described before, where the secretory product is water-soluble and lost during the normal tissue processing we use to examine sections of tissue stained with hematoxylin and eosin. Down below, if you look very carefully, you can see three profiles, a duct system. These ducts, remember, carry the secretion product from the secretory cells to an exterior location. And these locations can be often some distance from where the gland is housed within the body. And we'll see an example of that when we look at the pancreas. It has a long duct that opens into the duodenum some distance away. Well, when we look at large glands, we can see that they have a certain type of organisation. Here is a section through a large salivary gland. When you look at from this magnification, it's rather deeply pink staining. And you can identify, or you'll be able to identify, the different types of salivary glands because the three types of salivary glands and the pancreas have a preponderance of a certain sort of secretory product. Some are purely mucus, some, such as the parotid gland and the pancreas, are purely serous. Some are mixed, and using that criteria, you can distinguish the three different salivary glands and the pancreas. But all these large compound glands are divided into lobes and smaller components called lobules. Look very carefully at this section here, and you can see large connective tissue spaces dividing this gland into large segments. The septa of connective tissue are dividing this gland into lobes. Smaller segments of the connective tissue can divide the lobe into a lobule. So look carefully at this slide and you can see lots of tiny little divisions. They're separated by white spaces here, which is artifactual, caused by shrinkage of the tissue, but it's a good way of really illustrating that all these compound glands are divided into large components called lobes, and each lobe has subdivisions called lobules. And later on we're going to learn that ducts and also blood vessels access the secretory units, first of all by coming in through these large connective tissue spaces between the lobes, and then through the smaller connective tissue spaces going to each lobule and therefore to the secretory units. Let me just diverge slightly and introduce you to the myoepithelial cell. These are contractile cells. And as you can see here, if you look very, very carefully, this is a section through a sweat gland. And what you see, the paler regions, the paler clusters of cells, are the secretory units of the sweat gland. The small darker little tubes you see is the duct portion of the sweat gland. And I'll talk to you in more detail about these glands when we look at skin. But what I want to bring your attention to is the myoepithelial cell. As I said, they're contractile cells and they wrap around the secretory units. They hug the secretory units like an octopus. And you can see very, very fine little red lines on the outside surfaces of these secretory cells. They're the processes of the myoepithelial cells wrapping around the secretory units. Myoepithelial cells are very important in exocrine glands because they wrap around the secretory units and help with the secretion moving out of the cell into the luminal space. Now, sometimes glands secrete cells. They house stem cells, and the product of these glands are other cells to replace cells higher up in epithelial surfaces, above where the glands are. They don't produce proteins or mucus, they produce other cells. If we have a look at these three images, they illustrate secretory cells and also stem cells. This picture here on the left-hand side illustrates a number of finger-like projections that protrude into the lumen of the small intestine. These finger-like projections are called intestinal villi, and labelled here is a single intestinal villus. Where that label is pointing is to the underlying connective tissue, the lamina propria. But on the surface of these villus or villi is an epithelium, and that epithelium is involved with absorbing material from the lumen of the gut into various blood vessels and lymphatic channels that run in that lamina propria. But on the middle section, you can see some red stained cells that live in the glandular part of this epithelial surface, labelled here as small invaginations into the underlying lamina propria. That's where these secretory cells live, deep in the glands and not on the surface. Well, besides these secretory cells, there are also stem cells shown here. Different stains used to show up clumps of chromosomes in some of these cells that are undergoing mitotic activity. Well, these stem cells at the base of these glands produce other cells that move up along the surface of the little glands, and they differentiate into secretory cells that you see in the middle section, or they can differentiate into the surface epithelial cells and be involved in the absorption of products from the lumen. These stem cells are very important in epithelia because epithelial cells are continually being replaced, and they're replaced because of these stem cells being present in all the epithelial surfaces that we're going to come across when we look at the organ systems in later lectures. Let's look at how ducts are named in glands. Well, they can be named in a number of different ways. Firstly, you can name them according to the location you see these ducts. I mentioned a large compound gland earlier, and that large compound gland is divided by connective tissue into large lobes and then smaller lobules. Well, histologically, you don't often see sections through ducts that are in between the different lobes into lobar ducts. You only really see the small ducts. And those small ducts, when they're located between a lobule, is called an interlobular duct, and within the lobule, it's called an intralobular duct. You need to be able to identify these different ducts when you see sections through some of the glands. Often, they're classified according to not just their structure, but also their function. They're called excretory ducts if their job is only to convey the secretory product. And most of the large ducts, the interlobar ducts, are excretory ducts because they have no other function than carrying the secretion product to the location where the secretory product is going to be active. But a lot of the secretory ducts are involved with changing the compounds, the components of the secretory products. They change the compounds, the components, because they're involved with transport of material into the lumen and the absorption of material from the lumen. So they all have a secretory function, just as much as the secretory cells do. Absorption of material or secretion of material. And in this case, you will see in glands often ducts we term striated ducts or intercalated ducts, particularly in serous secreting or protein secreting glands. Remember, these striated ducts and intercalated ducts are really involved with modifying the secretion product. Sometimes we name ducts according to where they're located. For instance, the ductus deferens in the male reproductive system. Sometimes they're named after the individuals who first described them. Well, let's have a look at a couple of images illustrating certain ducts. Here's an interlobular duct. Notice the secretory acini, or the secretory units, on the right-hand frame of this slide. Secretory units are serous because they're rather dark staining, pink staining. They're the granules inside the cells at the apex of the cell, the protein granules. But look at the duct. It's separated from the secretory units by a fair bit of connective tissue. So that indicates that that connective tissue is dividing the gland into different lobules. Down below, that structure that has a reddish colour in the middle happens to be a blood vessel. Normally, as I said earlier, blood vessels and ducts accompany these connective tissue septa, separating lobules from each other. Now have a look at this section. It's taken through a serous secreting gland. What dominates are the white circular spaces you see. They happen to be fat cells or adipocytes. And similar to other mucus secreting cells I explained earlier, the content of these cells is lost during processing. Have a look at the left-hand side of this picture and see if you can make out intralobular ducts. They have a circular shaped lumen, and if cut nice transversely, then they have a nice cuboidal epithelium. This is a higher magnification, again through a protein secreting gland, showing striated ducts. Again, look at the tube on the left-hand side of the image. It's a striated duct. But have a look at the luminal area and then move your eyes and look at the basal area of this epithelium. And notice that there are these little pinkish or reddish stripes running along the basal aspect of this epithelium. That's why they're called striated ducts. And those striations represent, firstly, the arrangement of mitochondria in columns along the basal portion of the cell. And these mitochondria are arranged within many, many basal folds of the epithelial cell. And these mitochondria are there to provide the energy for active transport, because striated ducts are very important in protein secreting glands because they modify the secretory product. So these mitochondria provide the energy for active transport to move material from the lumen into the underlying connective tissue. And the basal folding is a way of increasing the surface area to create more space for these transport channels and also to house many, many more transport proteins. Now have another look at this slide very carefully. It again is a serous or a protein secreting gland. Pick out the secretory units or secretory acini. An acinus is a grape, a berry-like structure. It's a cluster of cells. But see if you can find an intercalated duct. They're very hard to see. They're very, very, very small in dimension and very short, and really only obvious in protein secreting glands. Not mucus secreting glands, because in mucus secreting glands the secretion product is not modified. But in protein secreting glands, as I've indicated earlier, it is modified. So these ducts are also quite prominent when you see sections of protein secreting glands. Well, have you found it? There it is there. Little tiny white space is the lumen, and if you look very carefully you see a cluster of cuboidal type epithelial cells surrounding or making that lumen. That's an intercalated duct. Well, ducts have many functions, which I've explained as we've gone through this lecture. One, it's just to convey secretions, and we call it an excretory duct. Sometimes the duct needs to modify the secretion product of the gland, and we'll see examples of that when we look at sweat glands in another lecture on skin, and also when we look at the salivary glands. Sometimes ducts store secretion. When we look at the prostate gland in the male reproductive system and also the mammary gland, we'll learn that when the secretion product is produced by the cell, it's often stored in the duct system until it's used or wanted. Well, now let me explain the term mucosa and also serosa. Remember when I showed you the image of the stomach mucosa, I asked you to remember that word, that term, mucosa, because in subsequent lectures, I'm going to use the term mucosa and also serosa quite often. So it's very important to understand what I mean by these terms. And they relate to glandular epithelium. They relate to the glands that I've just been describing, the exocrine glands. Well, you're familiar with this section on the left-hand side. It's a section through the intestinal villus, and it shows the epithelial surface sitting on the lamina propria. Remember, the intestinal villus is a finger-like projection that protrudes into the lumen of the intestine. Well, it's actually an example of a mucosa. A mucosa lines cavities that are connected to the outside of the body, such as the intestine. We'll see mucosas also in the respiratory passages and in the urogenital system. Again, epithelial surfaces that line cavities exposed to the outside of the body. So when we look at an endothelium later on, or the epithelium lining a blood vessel, we don't call it a mucosa. Why? Because blood vessels are not connected to the outside of the body. On the right-hand side, this is a very low magnification of a section through part of the small intestine. And those long extended processes are actually gross anatomical components of the small intestine. And where the label of mucosa is, shows you these villi under very, very low magnification. But remember, the villus, the epithelium, and the underlying lamina propria, plus sometimes a little bit of a little muscle layer you might see there, is called the mucosa. It's facing in towards the lumen. It's a cavity of a tube that's connected to the outside of the body, which is what the gut is. Here's another example of the mucosa. This is the stomach mucosa that I asked you to recall earlier on in the lecture. So now you know what the mucosa means. It's labelled here under low magnification on the right-hand side. Underneath it's supported by what we call a submucosa. And I'll talk about that term later on when we look at the description of the structure of the gut wall. But again, it's just to emphasise that the mucosa is the surface epithelium and supporting connective tissue or lamina propria, which you don't see a lot here because the mucosa is dominated by secretory cells. But it's facing the lumen and it's in a tube that's exposed to the outside of the body through the esophagus and the other parts of the gut. Well, let's look at a serosa. A serosa is an epithelial surface that lines cavities of the body that are closed. They're not exposed to the exterior of the body. Cavities such as the peritoneal cavity, the thoracic cavity, the pleural cavity, the pericardial cavity, all those surfaces are lined by what we call a serosa, a very thin, squamous, simple squamous epithelium that's found on the surfaces of these cavities. On the right-hand side is actually part of the large intestine or the colon. If you look inside at the lumen and you see the many folds stained very purple in this section, that's the mucosal surface in contact with the lumen. It's a mucosa because it's lining the cavity of the gut here, which is exposed to the outside of the body. But on the other side of the gut wall, the serosa is exposed to the abdominal cavity, the peritoneal cavity. So that's why it's called a serosa. When I lectured about epithelium, I mentioned that one function of simple squamous epithelium is to act as a lubricating surface. Well, often in these cavities, there is a serous secretion, a watery secretion that helps lubricate these cavities and helps create a friction-free environment, because often these cavities have contents moving all the time. The lungs increasing and decreasing during breathing. The pumping of the heart inside the pericardial cavity requires a friction-free environment, a surface that's lubricated by these serous secreting membranes or cells. Well, in summary then, make sure you understand what a gland is. It's a secretory unit derived from epithelial cells. Make sure you're aware and can identify the difference between serous and mucus secreting acini, or little clusters of secretory cells, and mucus and serous secreting glands. Know the different ways in which the secretion product is delivered to the luminal surface, and know how glands are named, simple or compound, and how they're classified by the way they appear, either unicellular or multicellular. And finally, know and appreciate that ducts are very important in some glands because they can modify the product. In other glands, they merely convey the product to the surface or to where the secretory product is active. So I hope you enjoyed this account of exocrine glands. I hope you now know something about these glands, and I hope you also look forward to learning in a later lecture about endocrine glands and also paracrine glands. So thank you very much for listening.